in the next couple of minutes. But Jera, if you want to start, okay, I see you started recording. Okay, so hey everyone. Today it's my pleasure to introduce Dr. Andrei Lupas. He is the director of the Department of Protein Evolution at the Max Planck Institute in Tübingen. And so he sort of started his scientific career actually in the States, where he completed his PhD in molecular biology at Princeton, after which he traveled back to Europe and completed his postdoctoral training at the University of Munich and at the Max Planck Institute of Biochemistry in Martinsried, both in Germany. And then after that, he came back to the States and served as the assistant director of bioinformatics at GlaxoSmithKline in Collegeville, Pennsylvania. And after that, since 2001, as I mentioned, he's been the director of the Department of Protein Evolution at Max Planck Tübingen. And so he's sort of been in this space of protein evolution and bioinformatics for some time, and has published some pretty landmark papers where he's integrated both. For example, in 2015, he and his team reconstructed a sort of vocabulary of ancient peptide fragments that may have led to the emergence of more complex folded proteins that we see today. And now his group attempts to understand how this emergence may have played out using techniques like crystallography, cryo-EM, and NMR. And then he's also made a major contribution to the development and improvement of the MPI Bioinformatics Toolkit, which a lot of you may have used. It's sort of this integrative web server where you can work with large protein sequence data sets, identify homologous protein sequences, do some homology modeling, and essentially do complex analysis and visualization of relationships in large sequence data sets. And lastly, he actually served as an assessor for the critical assessment of protein structure prediction competition. That is sort of this competition that brings together groups from all around the world who compete to see who can best predict protein structure. And that's been going on since 1994, and Dr. Lupas in 2015, as part of the 14th installment of CASP, was an assessor, which was actually that particular year was the competition where AlphaFold won by quite a wide margin. And so Dr. Lupas is an expert in protein evolution, also in protein structure prediction, and in studying the nature of protein structure and function relationships. So we're really excited to have him speak here today. And I should also note that it is 5:30 p.m. In Tübingen, where he is right now. And he said it's no problem, but still we really appreciate him speaking around this time. So everybody, please give him a virtual round of applause. And Dr. Lupas, whenever you're ready, you can take it away.
Sure. Thank you very much. And indeed, I will be talking exactly as you introduced on the potential of ancient peptides to produce folding as part of the coevolution of RNA and polypeptides. And so let me share my screen here. Okay, and let me go into projection mode. All right, and have the cursor. No. Oh, it's here. No, not here. It's here. Okay, and here's my laser pointer. All right, so I will tell you about our work in tracking the origins of folded proteins. I will try to convince you that this is an entirely non-trivial property of polypeptides, which is actually so rare that in polypeptides of any significant length, like 120 to 150 residues, we calculate that the proportion of folding polypeptides in a random set is less than 1 in 10 to the 20th, and probably so far less that it's only about 1 in 10 to the 50th, or even less than that. So the origin of folding in the protein world is definitely something that needs to be explained. All right, before I go into this, I just want to show you a few slides about where I am. This is the Max Planck Institute for Biology in Tübingen, where my department is located. And you see we're smack in the middle of things, which to us is a great thing. To my wife who loves the sea, it's not a great thing. So there's a long way to go to the North Sea. There's a long way to go to the Mediterranean. All right, so what do we do in the Department of Protein Evolution? We mainly study how the sequence of polypeptides specifies their structure to the extent that they have a defined three-dimensional structure, and how this structure enables chemical mechanism. How does structure lead to an actual chemical activity? And we start every project with a bioinformatic analysis, with developing hypotheses. Then we clone and express the proteins. Unlike most groups, we start every project with new proteins because we're trying to track a process and not understand a particular organism or a particular pathway. And then we characterize the proteins, we determine their structures, although nowadays, with the advent of AlphaFold, a lot of the structural work is already in the bioinformatic precursor to the project. And then at the end, we go into the living organism, often bacteria, but not always. We've already done experiments in fish, for example. And try to see whether the proteins that we think we understand, in fact, do the things we think they're doing. And the department has a fairly wide assortment of competencies ranging from bioinformatics, biophysics, NMR spectroscopy, crystallography, various forms of biochemistry, protein design, and we even have two emeriti. Joachim is 83 years old, Volkmar is 87 years old, but they're still in the department every day doing their work. And Joachim had a major breakthrough last year, which makes me very happy. Essentially, he answered a question that he had been pursuing for 40 years. And so I'm really happy that they're here because it illustrates to our students and postdocs that science is not a profession, it's a calling. We're scientists because we have this calling to find out things, and it doesn't stop with going into emeritus status. All right, let me start in trying to explain the problem to you. And this timeline shows you in blue here major biological events, and in black major geological events in the history of the Earth. And there's a variety of things to point out, such as, for example, that the biggest progress we had during the biggest catastrophes, so the oxygenation event, which released one of the most potent poisons of living beings, namely oxygen, coincided with a massive ice age, the so-called snowball Earth episode. And that in turn is very closely correlated with the origin of eukaryotes. And so we can discuss that to a great length, but I don't want to do that. What I want to do is to point your attention to this part of the graph, which shows you the time up to the last common ancestor. And what we know by now, based on a tremendous amount of primarily bioinformatic work, is that the last common ancestor was biochemically modern. Essentially, all folded protein forms, all prototypes had been invented. All the reactions in biochemistry had been established. If we saw cells from the last common ancestor population, we would think they're just prokaryotes. They're similar to the organisms we have now. And if you look at this timeline, you realize that if the heavy meteorite bombardment had happened, which we are not sure, geologists can't look that far back in time with any degree of reliability. But if this happened, then practically we had oceans starting from about 4 billion years ago, which means we only had a few, maybe at most 500 million years to evolve all the biochemistry we have. And that's pretty amazing to me, especially considering that folded proteins are not that easy to come by. And so if we look at a timeline of molecular innovation that led to the first living beings, we start with metabolic networks that by now seems pretty well established. And these networks have to give us polymers because we have to reach the biopolymers, nucleic acids, polypeptides, lipids, and sugars. And then at some point, we've had the first polymer networks integrated. And from that, we got autocatalytic replication. At some point, probably after autocatalytic replication, we had compartmentalization, which led to membrane-enclosed vesicles. It could very well be that these earlier forms were compartmentalized in different ways. For example, through differences in viscosity between the fractions containing the biological polymers and the abiotic environment. But then we still have not generated sequence-specific polypeptides. The emergence of the code comes after compartmentalization. It's pretty clear. And so many of these events come surprisingly late in the origin of life. And so first, I would like to dwell briefly on the fact that the RNA world was actually an RNA peptide world, because RNA can encode genetic information. It can catalyze reactions. It's a chemically active polymer, but it's limited in both activities. And proteinogenic amino acids were produced in the same reaction networks as nucleotides. And by now, particularly your colleagues at Georgia Tech have shown that they can be strung up into polymers abiotically and produce, especially if you allow for deoxypeptides, lengths of 10, 20 amino acids strung in a row, which allow you to do a fairly large number of things. Now, these peptides would have offered many potential benefits, and they would have done so without sequence specificity. Just by virtue of the amino acids that would have been incorporated and the various groups, they could have acted as chelators of metals and small molecules. They would have promoted RNA thermostability, which was a big problem. They would have started engaging in various forms of oligomerization, and so on. And indeed, many peptides are membrane active. So if we consider that cellular envelopes arose around that time, peptides would have been essential in order to functionalize any membrane that arose. So I think there's a ton of arguments why the RNA world was an RNA peptide world. But then the next question is, why aren't we in the RNA world? If you have the polymer networks, if RNA is sufficient to give you autocatalytic replication, then in essence, you would get to a living systems. You would have a system which is capable of Darwinian evolution, and therefore a system which is the first step in life. So why didn't we stay here? Why did we go on to the DNA protein world? And, you know, whichever way it happened, I think that this closed circle of RNA and peptides was already a life form. It already had the critical elements that are needed for the first life. So why go on from this? And basically, we know nature is a tinkerer, which means it doesn't invent new things because thousands or millions of years in the future, these will prove useful. If we made the transition to the DNA protein world, in my opinion, we did so because of a catastrophe. The question is, what nature did this catastrophe have? And I note that there's many arguments in favor of this meteorite bombardment. And there's also arguments about particularly heavy impact late in this time period, which is sometimes referred to as the late veneer, which may have raised the surface temperature of the Earth considerably. We know that RNA is by far the least stable of the biological macromolecules. And so any life form that relied on long RNA molecules for its genome would have been heavily disadvantaged or even gone extinct. And I can only assume that at the time already some organisms made the transition to DNA. And Patrick Forterre, the famous biologist who has worked on archaea for half a century, proposed that the transition to DNA was made as a resistance to phages, to bacteria, because viruses, because viruses and predatory forms are as old as life itself. So if we were an RNA life form, we certainly had RNA viruses to prey on these life forms. And so it's quite plausible that some organisms develop DNA as a resistance mechanism. And then when the impact came, they survived because DNA was not prone to the same issues. Whichever way the transition, the chemical transition from RNA to DNA is not problematic, but the protein folding problem is a huge obstacle. So what is the protein folding problem and why is it such an obstacle? First, let us establish that the chemical activity of proteins is the essence of life today. Everything in nature is done by proteins. Nucleic acids are only there to specify which proteins are made when, where, and in what amounts. Everything else is done by proteins, including the replication of nucleic acids, their biosynthesis, their modification, their everything. All right, and these proteins with chemical activity, they must assume a defined three-dimensional structure in order to exert catalysis. And we call this folding. Now, nucleic acids fold spontaneously and very easily. So if you do a PCR reaction, your nucleic acids fold and then reanneal, and then they, melt and then they reanneal. And you can do that as often as you want without significant loss of material, whereas with proteins, it's much harder. And in fact, it was impossible before we got highly thermostable polymerases to run this. And it's maybe not uninteresting to think back to the time of Gobind Khorana, who helped solve the question of which triplets encoded for which amino acid. He did it by an early form of PCR where they would purify the polymerase and add new polymerase every time, right? Now, you can do it if you have one of these big departments, but otherwise it's a bit difficult. All right. Now, as I mentioned to you, random polypeptide chains of any significant length have a minute, essentially negligible likelihood of folding. How do we know that? Well, there was a time about 20 years ago when several groups embarked on screening large random peptide libraries for folding exemplars. That was Jack Szostak and Andreas Plüchtun and Greg Winter and Michael Hecht. And they all got essentially either nothing or at best one or two folded exemplars. And so if you report that back to the size of their libraries, that's much less than 1 in 10 to the 10th, especially since their starting points in almost all cases were not random. They were already biased towards certain patterns of amino acids. Now, we know from many evolutionary studies, indeed most of them done computationally, that nature bypasses the folding problem by using a vocabulary of domains which can be shuffled and recombined and modified, and which yield the fact that, for example, a third of all your enzymes are Tim barrels. Why? Because it's a scaffold that can give you almost any activity. Almost half of all your hydrolases are alpha-beta hydrolases. Again, why? Because that was a scaffold that worked and you could get a very, very large number of activities mounted on one scaffold. And so domains are the units of protein folding today, but they're too complex to have arisen de novo. And so the question I started to work on when I came from SmithKline to the Max Planck Society was, how did these folded domains originated? Where do they come from? And my hypothesis was that there were peptides ancestrally which had evolved in the context of ribozymes, and they were optimized to form secondary structure because of the need for specificity in the interaction between the RNAs and the peptides in order to obtain catalytically active complexes. And so these peptides folded into secondary structures and super secondary structures using the nucleic acid as a scaffold. So what happened when these scaffolds went away? And what I think happened was that these fragments showed their ability to catalyze the folding of domains through a few simple mechanisms, which I will talk about, and in the process laid the seed to the first complement of folded prototypes at the transition from the RNA world to the DNA world. Right. Now, the hypothesis that I started with was, if this is true that I just showed you, we should be able to reconstruct ancient peptide forms by comparing the sequences of modern proteins. And the way I looked at this was from a linguistic perspective. And I'm going to show you here. It helped that my boss at SmithKline Beecham was David Searles, who was a computational linguist and who had cut his teeth on artificial intelligence, which now, 20 years later, has broken into this amazing flourishing, but back then had not really yielded that yet. And David had become well known in bioinformatics by showing that DNA sequence follows the Chomsky hierarchy, the Chomsky grammar hierarchy. And therefore, he was thinking very much like a linguist. And so he kept asking me, how do proteins behave relative to the Chomsky hierarchy? First off, I had no idea what the Chomsky hierarchy was. But second, as I learned what it was, I decided, no, not at all. Not at all. And I think that, or I thought back then, that had to do with the fact that proteins are not a code. In DNA, if you recode what triplets mean, you'll just get a new meaning. It's a code. With proteins, they're the real thing. They're the thing that does the chemistry. So it doesn't behave at all like a grammar. In fact, what I realized is proteins behave like vocabulary. And so I would like to illustrate this briefly for you. I'm going to take a very old word, kane, which we know from Sumerian cuneiform tablets. It was a very important word in that world because there was very little wood. And so the Sumerians made everything out of reeds, boats, huts, fire. All of this was made with kane, with reeds. And so, unsurprisingly, direct descendants of this word are found in all Semitic languages, also Hebrew, Arabic. And when the Greek came from the North Caucasus into the Peloponnese a few thousand years ago, they obviously didn't know, hadn't encountered reeds, and therefore took the word from the Semitic languages along with knowledge of this new plant, and they called it canna. Now, why the Latins took the word from the Greek, I have no idea, because as we know, Rome was founded in a swamp, which means they had plenty of experience with reeds. Why they took the Greek word, no idea. But they did. And so via the French canne, we got the English cane, which still means essentially the sugar cane is a reed that we use for making sugar. Right, and so this is the orthologous descent over many thousands of years, where the same meaning was preserved from the ancestry to today. But in Greek times, there were also paralogies that happened. The Greek noticed that the reeds were straight and had nice nodes at intervals. So they called the measuring rods they used canon. And from that, they also derived the abstract term of rule, which is canon as well. And from there comes the canon as the church rule, and the canon as singing by rules, as the musical form. And also in Greek times, the Greek wove wicker baskets from very young reeds, which are still quite flexible. And they called this canastron, which gave us the canister. And also a few other words like canasta, which is a card game. The Latins, as always, extremely practical, took the reeds. They noticed they're hollow. If you puncture the membranes, they're hollow. And they're tapered, which means one end is smaller than the other end. So you can push them into each other. And then you can make a pipe, a duct for water. And that's what they did. And they called it canales, from where we have the canal and the channel and the canyon, and also things like the canalure and the cannelloni, and the can, a cylindrical metal container, right? So you see that in essence, the last common ancestor of church rule and a TV channel was a Sumerian plant. And that is how proteins should be thought of, because the proteins that we define into structural groups are often very different in their functionalities. What they do have in common are structural properties, some, and catalytic properties, which depend on that particular structure. That's what remains after billions of years of evolution. And here I can show you such an example. If we start from this hammerhead motif, which is part of the vocabulary we defined, we can find this motif in a ribosomal protein, L27. This protein looks perfectly folded, but to my knowledge is not structured in solution unless you give it the RNA scaffold. Then it folds on the scaffold. But it is the ancestor of a soluble form which is folded autonomously. This is a subunit of the exosome. It still works on RNA, and it still has substantially the same fold, but it has become autonomous in its folding properties. Now, we see that somewhere very early, we had this paralogous event where this fragment was duplicated and formed a cavity between the two sides of this duplication. And this cavity turned out to be ideal to bind biotin. And so all the biotin using enzymes go back to this ancestral form, which invented the use of biotin in order to promote catalysis. And we also have a few other forms which continue to act on RNA. The biotin enzymes don't, these do. And they essentially preserve this fragment, but their functionality has changed quite fundamentally relative to this ancestor. Right, and we also have analogy, as we had with language. There are analogous forms where we can track through sequence comparisons and evolutionary studies that these probably are not related directly to these forms. They just happen to look the same way. All right, so what did we do? We went out into a structural classification. In this case, we use SCOPE, the structural classification of proteins, because we wanted to find fragments that were locally similar in sequence and in structure, but globally dissimilar. So we wanted to find pieces where we could make the arguments that they are homologous, and yet the environment in which they are used is completely different from entity to entity. And these, in our opinion, would have been these ancient peptides which predated folded proteins. And so here is an example, the helix hairpin helix motif. And you can see here part of the multiple alignment. The conservation is pretty obvious. In structure, the conservation is also quite obvious. But the environments in which this fragment occurs are completely different. In fact, this is one of the fragments we found which structural biologists had already defined a long time before we started this work. And that makes perfect sense. Humans are one of the most amazing pattern matching machines. And so the fact that people who have dealt in detail with the structure of these things notice the similarity across different folds, to me, seems absolutely obvious. And so the motivation of our study was not to find only new forms. It was to find as many as we could substantiate. And the more of these we found that others had seen individually before, the more we saw a justification that we're on the right track. And in fact, it helped us get this past reviewers because they said, oh, okay, we all accept that, for example, the Rossmann fold is a homologous structure. And so if you find the Rossmann strap loop as an ancient fragment, that makes sense. You should find it, right? And so by the same token, I would say we should have found the helix hairpin helix motif because by all accounts, people noticed that this had similarity in sequence and structure across proteins with vastly different folds. And so in order to get there, we built the bioinformatics toolkit. In essence, I built the first toolkit when I was at GlaxoSmithKline. I couldn't take it with me, so I built a new one when I arrived in Tübingen. And Johannes Röding, the originator of HHPRED, while he was a postdoc in my department, was also at the root of making the first version of the MPI toolkit. The toolkit is very popular. It's used for a variety of tasks, not least for teaching classes. We see that all the time from the queries we get and from the fact that we sometimes have tens of the exact same job being submitted from the same IP number, which suggests they're students sitting in a class and having this assignment. I must admit, I work mainly through the toolkit, and that's why it has the functionalities. It has because we develop tools and put them in here so that our entire department can use tools, even though they're at the experimental side and not the computational side. So anyway, that's the toolkit. And with the help of the toolkit, this is the vocabulary of fragments that we published. Now, one massive shortcoming of this is I didn't realize it. We didn't put 42 fragments here. We just put 40. It would have been trivial to just expand our cutoffs in order to include 2 more, because then we would, of course, have had the answer to life, the universe, and everything. And I can't explain why I didn't do 42, but I didn't. So anyway, what you see here are a number of annotations. I won't go into all of them, but you see that we have a considerable enrichment of fragments that interact with nucleic acids. We have some fragments that interact with nucleotides. You'd say, well, 2 and maybe 3. But I say these 2 span the by far biggest families of enzymes in nature. One is the Rossmann structure, and the other one is the P-loop of NTPases. So basically, there are only 2, but they span an enormous range of protein domains. And all these things here, we don't know that they don't interact with one or the other or with metals or something. All we know is that in the crystal structures we have at our disposal, they could not be seen in direct physical contact with nucleic acids, nucleotides, or metals. That's it. All right. So you see all these dotted lines here. These are the fragments which make folds by repetition. So the absolutely dominant phenomenon we see here is repetition. Repetition is at the root of at least a quarter, probably a third of all domains we see today. And we also see the same proportion when we go to the level of fragments. Repetition is huge. The other thing we see, which Greg Winter already described, is decoration. The fact that you have a core which knows what it's doing, and you just give it peptidic material, and this material will arrange itself around the core. What we do not see, and I was completely unprepared for this because I thought that would be the dominant mechanism, but it's not, is a recombination of these fragments. We don't know any domain which contains more than one type of fragment. The fragments don't like each other, right? And that was really surprising. I won't have time to go into that, but we've tried very hard, and we've only gotten one of these Lego combinations working after we mutated a third of the residues in the fusion protein. So that's not plausibly something nature would do. So right now, I would say, as far as we're concerned, recombination of fragments is not a phenomenon that leads to folding, whereas repetition is. And so let me go through the ribosomal RNA part of the talk and go directly to an example of repetition. This fragment right here, fragment number 28, it is at the origin of a large number of folded proteins, of which one is repetitive. It's the TPR repeat, which I trust you're all familiar with. All the other forms of this hairpin are not repetitive. This one is. And so, since it also occurs in a ribosomal protein, we asked whether we could take the hairpin from the ribosome, amplify it 3 times, and get a TPR repeat. In some sense, it should work. In another sense, it should absolutely not work. Why would we get exactly the interface that TPR repeat has if we take a hairpin that's not optimized to have any repeating interface? So we did it. And lo and behold, here you see the crystal structure we got in cyan and the thing we expected in gray. We pretty much got a TPR repeat just by repeating a hairpin from a ribosomal protein 3 times. Now, we could show spectroscopically that the repeat itself was most likely at least partially folded, but we had to introduce a minimum of 2 mutations at the interface because the interface was not hydrophobic enough to allow for folding. But it worked fine. And now with our design capabilities, I'm pretty sure we could design this such that we can then go in nature and find a version of the hairpin that will fold without us mutating anything. All right. We also took another one of these ancient fragments, the so-called propeller blade. And this is a fragment at the root of propellers, of course, but also of beta prisms, and of a protein which is neither a prism nor a propeller, but a strange protein mimicking MHC, the binding site of MHC receptors. And yeah, our question was, can this be ongoing since these are all repeats? Did these things happen once and then from then on we evolved by differentiating the prototypes that were there? Or can we actually build new exemplars all the time by repeating this ancient fragment? And so we had this natural sequence which had nearly identical blades, and in some cases fully identical blades, with a very small number of changes in the amino acid sequence, which we could map to the crystal structure and showed that they were almost all around the binding site, the predicted or expected binding site. But further than this, we also saw in the DNA that there were no synonymous mutations. Everything was non-synonymous, which means doing dn by ds would not have been meaningful here because ds is 0. Well, no, ds is infinite. S is 0. There's no synonymous mutations. And so in essence, this looks like something that evolved yesterday and is under infinite selection pressure, which of course it isn't, but looking at the history of the strain where we found this gene, we expect that this gene is less than 100 years old. And if it acquired all these point mutations, it suggests that this thing, which is the receptor of an immune-like protein related to the human apoptosome, we think that this might be a sensor for phage proteins, the phage capsids, and that therefore the mutations are racing to become of higher affinity to the phage capsid. But it's a pure speculation. We have no actual concrete evidence. All right, but what we did do is we took this blade, which is highlighted in red, and looked at whether we got folded exemplars by repeating it. Now, if we didn't repeat it, if we just took this blade, it didn't fold. It was very soluble, very friendly, but it did nothing structured. But then as soon as we put 2 copies of this in one construct like this, we got this 8-bladed propeller, which is a tetramer of 2-bladed monomers, right? And so then we put 3 copies of the blade in a construct, expecting that we can't make an 8-bladed propeller from this. The parent is, by the way, 7-bladed. So that's a prime number. All right, so we put 3 and we got a 9-bladed propeller. And you see it has a slightly triangular shape because the interfaces are not the same. And this then, of course, led us to put 4 copies of the blade into one construct. And we got an 8-bladed arrangement again, but you see the tension now accumulates at the interfaces here. So whereas this looks like a 7-bladed propeller, the interfaces here and here accumulate most of the geometric tension that arises. And then, of course, you can ask what happens if we put 5 blades in the same construct. It was a dimer, so I held out for a 10-bladed thing. But in fact, it wasn't even a propeller. It looks like a propeller, but a propeller is like this. This is like this. One half has switched sides and goes in the opposite direction. And the tension here is compensated for by one blade becoming a helix and plugging the hole. So if you look at this, one is a 5-bladed piece, the other one is a 4-bladed piece. And the C termini in this case are next to each other rather than on opposite sides as in all the other constructs. And so if you look at it from the side, you see how the helix plugs the hole. So this is what Richard Goldschmidt, writing about developmental biology, called a hopeful monster. Relatively minor genetic changes lead to a macro change in phenotype. And so I call this a hopeful monster. It's stable, it's friendly. We don't see anything like it in nature, even though nature must have encountered it many times, which means it's probably not very useful. But it's not hard to get. All we change is going from 2 or 3 or even 4 blades in one construct to 5 blades in a construct. Small genetic change, and the phenotype has changed substantially. Okay, I could go on and on about this, but let me just highlight we've done this kind of work with 6 of our fragments and have found in all cases that we can evolve towards folded structures quite readily. It's not a big problem. And now we are trying to understand decoration, because recombination we've looked for over a decade by now. We don't find any evidence anywhere. Whereas for amplification, I just show you this propeller, which clearly arose recently in evolution. For recombination, we see no evidence in the genomic data set. All right, let me leave you with the following thought. We can't travel back in time, which means all I've shown you is that it works this way. Whether it actually happened this way, I have no idea, right? All I can show you is that these mechanisms are so simple that they most likely happened many times over, right? So in some sense, we're playing this connect the dots game. Except, and right, you connect them correctly and you get a nice picture. The problem for us is we don't get the numbers. So we constantly ask ourselves, which dots should we connect? And most of our dots are extinct. And so if we connect the dots, we may come out to a completely different picture from what the starting picture was. And so with this, I would like to leave you on this thought. I have confidence that we can make statements about the statistical relevance of certain events, but their directionality and these sort of evolutionary scenarios, I'm deeply skeptical of. I think too often we're risking to have a picture that's completely different from that was there ancestrally. And in part, that also has to do with the fact that, as I said, nature is a tinkerer. It changes things randomly and then tries to get something that works better than before. It doesn't plan, it doesn't design, it just does. And so using Occam's razor in order to get these beautiful linear pathways going from form A to form B to form C to form D is absolutely not what nature does. Nature oscillates around form A and then maybe jumps over to form B. Sometimes it backtracks back to form A. Sometimes it goes on to form C. There's a lot of cycles happening in these, which we cut away because Occam's razor says if you don't have evidence, you can't postulate it. But now with AlphaFold, we're starting to be able to postulate a lot of things that maybe aren't solved by high resolution methods, but can be predicted with AlphaFold. And in fact, we can even predict the virtual sequence space around the form and see what the landscape looks like and what paths we might have in this landscape in order to arrive at new solutions. Okay? All right, so enough talk. Thank you for listening to me, and I'll be happy to answer questions.
That was great, Dr. Lupas. That was a really interesting talk. So yeah, we do have some time for some questions. So feel free to speak up or ask in the chat.
Hi, can I ask a couple of questions?
Sure.
This was wonderful. I really enjoyed this. But I want to raise a question about two things, particularly recombination as a role for moving things around genetically, interorganismally even, is finally getting its moment in the context of most genetics being thought of in a gradualist mode, one new nucleotide every million years. And the way you're treating, or the picture that you're presenting of the almost saltatory evolution of the modules of domains and their relationship with each other sort of suggests that that would be consistent with such domains traveling genetically on DNA, possibly on RNA, but in DNA coding protein, might involve mobile genetic elements. Is there any consistency or any examination of the possible association between the domains and their evolution as carried by or connected with mobile genetic elements?
I mean, these are excellent points. First, I misspoke. When I say there's no recombination, what I meant is there's no recombination of the building blocks. Because recombination underlies also repetition, it underlies decoration. Genetic recombination is everywhere, right? And so, for example, unequal recombination as the basis for amplifying repeats or shrinking them. Mobile elements, yes, in my opinion, the RNA world was all made of mobile elements, because there was no organismic specificity in any modern meaning. And so what we call the last common ancestor was a huge pool of genetically interchangeable forms in which some had this part of the information and some had another part. And so, yes, definitely mobile elements are at the origin of life, as far as I'm concerned. Which doesn't necessarily say that the forms we see today as mobile forms are directly related to any of these ancient forms. I think that, like viruses, this behavior can evolve multiple times out of precursors that didn't manifest either mobility or predatory behavior like viruses do.
Okay, well, just quickly, the second question has to do with, would you be open to considering a more flexible consideration of RNA as a carrier or perpetrator of modules in light of the recent Nobel Prize about microRNAs?
Oh, no, actually, no. But I would consider what Nick Hudd at Georgia Tech has been publishing for 20 years, which is that the original bases were not RNA at all. It was a completely different set of nucleobases, which then gradually converged on the RNA as a very late form of this. And I think he has quite a number of very interesting arguments. I also think that coming from a DNA-centric world, we completely underestimate the role and influence of RNA modifications, which are front and center to what RNA is. And we neglect them because we think of RNA as a sister form of DNA. But in fact, RNA, with all these modifications, can do a lot of things DNA does not.
There's a lot of more degrees of freedom than DNA by a lot. I mean, getting rid of some of those degrees of freedom made DNA more stable, I mean, a more stable nucleotide to have like evolution.
Right.
So thanks a lot. This has been fun.
Dr. Cunnin?
All right, thanks. Again, thank you so much, Andrea. It was a lot of fun. I really enjoyed that. So my question is also related to the previous question, but more from the biochemical side of things. So with regard to this helix, turn helix motif that you showed for both RNA and DNA binding, if you were to synthetically sort of build one of these motifs and analyze interactions with DNA and RNA, would that be more promiscuous, the most ancestral form? Or have you done those types of experiments? Would you mind
No,
sharing some of your thoughts?
I have not, but as it turns out, Jack Szostak did.
Okay.
Not on purpose, but in essence, he took where is it? The treble clef finger, which binds nucleotides and also nucleic acids. It's a zinc finger, and therefore it should be down here. This is the treble clef finger. So he didn't take it. He evolved from a random peptide library, and that was indeed a random library. He evolved something that bound ATP. And this ATP binder, it turns out, has converged on the treble clef structure. And in fact, because it has the cysteines at exactly the right places in order to form the structure, you can even find intermediate homology, sequence similarity. We know that it cannot be homologous because there was no treble clef finger, or indeed no natural sequence in the starting set. But the fact that it converged, and because of the dominant role of these cysteines, it gives an intermediate probability in HH-pred, alerts us to the fact that once you have converged to a certain point, if a few residues dominate your sequence, you're quite possibly going to look statistically like a potential homologue, when in fact you're not.
That's great. So my second question is more of a comment with regard to your slide on, comparison between protein sequences and natural language. You showed this really nice example with the word. And if you see, actually that made me think about the origins of my name, which actually comes from a Sanskrit word. And so I didn't see that in your slide. So maybe thinking about, because Sanskrit is apparently more, it sort of predates both Latin and Greek. So maybe worth thinking about adding that as well in your slide.
Oh, there's many more arrows that I could draw in here. The problem with that is that the slide then becomes, how should I put it? I can't communicate.
Sure. No, I was just
slide. But yeah, I have, in fact, the first author of the paper we published on fragments, Vikram Alva. Although he comes from a Tamil background, he also pointed out various Sanskrit relationships. But I didn't put them in because I wanted to keep it sufficiently simple to be able to explain things. Yeah.
Thank you.
Sam, you want to go ahead and ask your question?
Yeah, thank you. That was really cool. I'm more in the carbohydrate realm, so I was just learning some pretty basic stuff today. But just to clarify, I knew what a domain was coming into this, but is a fragment and a domain the same thing or not?
No, the domain is defined as an autonomously folding segment of the polypeptide chain. Now, we know of domains which have evolved not to be autonomously folding anymore, but we can always track them back to entities that are capable of autonomous folding, whereas the fragments are defined as subsets none of the fragments can fold by itself. They need help.
Okay.
They need either an amplification or decoration material that arranges itself such to help the fragment generate a hydrophobic core.
Okay. Glad you clarified that. And then you're saying or fragments don't like each other, and I guess I got confused about recombination. Like, does that mean actually, like, let's say you have two fragments and I don't know, you split both of them right down the middle and then you swapped their halves? Is that what you mean? Or actually mixing fragments in the same domain or something?
Yeah, what I mean is the second. What if I could take this piece and make a new domain by combining them?
Okay,
Right.
gotcha.
And we've never seen it.
That's really fascinating, yeah.
There is a protein which has the helix hairpin helix motif and the TPR hairpin. So we thought, oh, we just have to find two instances that happen to be compatible, and then we get a folded domain. Allow me to switch on my light. No.
Could I get in a question about metals?
Okay. All right. So basically, we tried to combine these two fragments, and we got a folded protein after we mutated about a third of the residues in the fusion protein. And so we did not take that as evidence that this process could happen in nature.
Cool. Thank you.
Right. And I'm just going to ask one question, then I'll divert it to you. And I think after that, we're going to have to let Dr. Lupas go. But
Sure.
yeah, so Dr. Lupas, I had a question about the helix, hairpin helix motifs so when you were searching for local motifs across different folds, I mean, how did you determine the size of the motif you were going to search for? Because I imagine the smaller the motif you look for, also the more likely it is that you could have had a random assembly of those specific amino acids together. So, I mean, did you use like an optimizing algorithm or did you like manually?
Well, we used HHPRED. In fact,
Okay.
HHPRED was built for this study. The fact that it's useful for other things is lovely, but it was built specifically for this study. And the shorter your fragment is, the bigger the likelihood that you're going to find a match, but the smaller the significance of this match. And so HHPRED intrinsically makes an optimization. It's not as greedy as SciBLAST, which tries to move beyond the ends. It has specific modules that prevent it from acting greedy, but it does try to optimize the length of the fragment in which you get the best overlap. And so basically, without any structure input into our starting bioinformatics, we found that HHPRED already pretty much gave us the boundaries of these fragments, which to us was an encouragement, right? Yeah. I had not mentioned that, but all the ones that have these black dots, they're all fragments that had been noticed individually by structural biologists before and reported in papers. So Vikram was kind of disappointed that so many were reported before. I told him, Vikram, this is brilliant. This is exactly what we were hoping to get, because we can show that through this approach, we can recapitulate everything structural biologists did individually. The important question for me was there any fragment that structural biologists proposed that we didn't see in our data set? And it turns out one fragment is right at the edge. It's the next fragment down from our cutoff. So everybody else is in the main set, which suggests to me that we're in the same direction as structural biology has been for 40 years.
Great, thanks. All right, and go ahead and ask your question, and then we'll end it there.
Okay, just quickly, in that pink patch there of the metal binding ones, are there any consistent associations of the type of metal or the role of the metal, catalyst or electron schlepper or structural, in the folds? Or this one does zinc and it holds the protein together, or this one does iron and it's schlepping electrons and stuff.
Right. As far as I recall, 3 of these coordinate iron sulfur clusters and 3 coordinate zinc.
Okay, so the guys that you're looking at are only ones that do zinc and iron?
Correct.
No copper.
No. We don't see any evidence for ancestral use of these rarer metal forms. It's essentially iron sulfur or zinc.
And the iron, this is a distributed iron sulfur, and then the zinc are structural. Well, zinc, yes. I mean, zinc can also be catalytic.
But in this case, we anticipate that they are catalytic, the zinc ions.
Okay, thanks. Thanks a lot.
Sure.
All right. We'll let you go, Dr. Lupas, but thanks so much again. That was a really nice talk, and yeah, it was nice to hear about a more biochemical perspective to some of the informatics works that you did. So it's nice.
Right. Yeah.
Thank you.
Well, by training, I'm a molecular biologist, right. But in fact, my PhD is in biochemistry. It's just that the department was called molecular biology.
Oh, I see.
Right. On chemotaxis, swimming behavior of cells and how they transduce the signal.
Okay, cool.
Yeah. All right.
All right.
Thank you.
Yep.
Thanks much.
Where am I? Here. How do I stop this?
And then Dr. Lupas, we actually have Dr. Alva speaking on December 6th. So
okay.
Yeah,
cool.
So I think that'll be a nice compliment to your talk as well. So
yeah. Well, maybe then I